Hello everyone. In this video series, I have already discussed in detail about the monohybrid cross of Gregor Johann Mendel and two different laws of inheritance which Mendel proposed on the basis of result of this cross. The laws are first law of inheritance, which is also called as law of dominance, and second law of inheritance, which is nothing but the law of segregation. Okay, but after performing monohybrid cross, Mendel think about the dihybrid cross, because we know that in case of monohybrid cross, he only studied the inheritance pattern of single character from parent to progeny. That means from one generation to the next generation. And in this cross, Mendel obtained consistent result. Okay, he got phenotypic ratio 3:1 and genotypic ratio 1:2:1. So one question which always come in his mind is that whether these two ratios will going to remain same if we consider multiple character at a time, because in monohybrid cross we actually consider pattern of inheritance of single character at a time. But if we consider multiple character at a time in a cross, whether these ratios will going to remain same or they change. To find out the answer of this question, Mendel planned next type of cross, which is nothing but the dihybrid cross. So in this video, I am going to discuss in detail about the dihybrid cross and how exactly Mendel performed this cross. Okay, on the basis of the result of the dihybrid cross, Mendel proposed his last or third law of inheritance, which is also called as law of independent assortment. Okay, so now we are going to talk about procedure of dihybrid cross. Actually, we know that in monohybrid cross, he studied inheritance pattern of a single character. That means he considered only one character of the organism, and then he focused on that character, and then he consider how the character is transferred from one generation to the next generation. Okay, so now in case of dihybrid cross, actually the inheritance pattern of two characters are studied simultaneously. Okay, so it is the type of a cross in which the inheritance pattern of two pairs of characters are studied from one generation to the next generation. So it is called as dihybrid cross. Okay, so to perform or to carry out this cross, the Mendel select two different type of plants. Okay, garden plant, and then he performed the experiment similarly that of the monohybrid cross. Okay, so we know that in case of monohybrid cross, Mendel used three different steps. So in first step of monohybrid cross, Mendel select pure parents. Okay, and then after selection of the parent, he perform artificial cross in the parent and then obtain F1 generation, and then after selfing F1 generation, Mendel get F2 generation. So these are the three important steps of classical cross which performed by Mendel on garden pea plant. Similar three steps were used by Mendel for dihybrid cross. So in the first step of Mendel's dihybrid cross, he select pure parents. Okay, so first parent of the Mendel was yellow and round seed pea plant. Okay, while the second parent was green and wrinkled seed pea plant. Okay, so these are the two different type of parents which Mendel select. This yellow is the color of seed and round is the shape of seed. Similarly, in this parent, green is the color of seed and wrinkle is the shape of seed. So in this case, Mendel select two character at a time. Number one, seed color, and number 2 seed shape. Okay, and because of these two characters, we can call this as a dihybrid cross. Okay, so in the first step, Mendel select pure parent, that is yellow round parent and green wrinkle parent. How he know that the parent is pure? And for that purpose, Mendel allowed selfing in these two parents, and then he continued three generations, and then he found that yellow round produced yellow round type of plants and green wrinkle produced green wrinkle type of plants up to 3 generations, and due to which Mendel conclude that both these are the pure parents. Okay, now this is the phenotypic characters of the parents. Okay, so now as they are pure, their genotype is yellow round plant have capital Y, capital Y, capital R type of genotype. We have discussed about the genotype in detail in monohybrid cross. Okay, this capital Y is the pair of allele which is responsible for yellow color in the pea plant, while the capital R is the pair of allele which is responsible for round shape of the seed in the green garden pea plant. Similarly, in case of green wrinkle plant, small y is the pair of allele which produce green color in the seed, while small r is the pair of allele which is responsible for wrinkle seed. Okay, if I draw the diagram of this seed yellow round, in this case you can find the plant produce round seed and the color of the seed is yellow, while in case of green wrinkle type of seed, plant produce green color seed and the surface of the seed show wrinkles. Okay, Mendel was aware about the fact that yellow color is dominant over green color and round shape of the seed is dominant over wrinkle shape. Okay, so this is the dominant character and the green and wrinkle is the recessive character. Okay, so this is the first step of the Mendel's dihybrid cross, that is selection of the pure parent and obtaining a pure line by selfing this parent up to 3 generations. Now, in the second step, Mendel performed emasculation and artificial cross. Okay, so this is the second step of the dihybrid cross. In this case, Mendel do emasculation of the female parent and then he transfer pollen grain from the male parent or male flower onto the female flower. Okay, so out of these two parents, Mendel select yellow round plant as a female parent and green wrinkle plant as a male parent. Okay, so now during the emasculation, Mendel grow both these plant and then he remove the anther of the yellow round flower before pollination. Okay, he remove the male gametophytes. Now, then he transfer the pollen grain from the green wrinkle flower to the stigma of female yellow round flower. It is called as artificial cross. We have discussed in detail about the artificial cross in monohybrid cross experiment video. Okay, so after emasculation and artificial cross in between these two parents, Mendel got F1 generation. Okay, so what is the phenotype and genotypic ratio of the F1 generation? Here you can find this yellow round type of a parent produce single type of a gamete, capital Y and capital R. We know that this gamete segregate, the allele pair segregate to produce gametes. So this capital Y produce single type of gamete, that is Y, and capital R produce R type of gamete. Similarly, this green wrinkle plant produce small y and small r type of gamete. Okay, this is the small y, single y and small r type of gamete. Now, these two gamete fused in F1 generation. So, the genotype of the F1 generation become capital Y, small y, and capital R, small r. So this is the F1 generation. And now in F1 generation, this is the genotype. The phenotype become capital Y is dominant over small y and capital R is dominant over small r. So the seed become yellow and round. Okay, so this is the genotypic and phenotypic ratio of F1 generation. Okay, so this is the first two steps of dihybrid cross. In first step, Mendel select two pure parent, yellow and round as a female parent and green and wrinkled seed as a male parent. Then he perform emasculation of female parent and transfer pollen grain of the male parent onto the female parent flower, and then he do artificial cross. The female parent produce capital Y, capital R type of a gamete, and male parent produce small y, small r type of a gamete. After artificial cross, the F1 generation seed were produced. The seeds show capital Y, capital R, small r type of a genotype, which is the yellow and round type of a seed. These results are similar to that of the monohybrid cross type of a result. Okay, in monohybrid cross, if we cross tall plant with a dwarf plant, in F1 generation, Mendel got all tall plant. Okay, similarly here, Mendel got all yellow and round seed. Green and wrinkled seeds are not produced in F1 generation because of law of dominance. Now, in the last step of the dihybrid cross, Mendel grow or use this F1 generation plant seed, and then he raised the plant, allow selfing in F1 plant, and then he collect the seed and raise F2 generation. So the next step or the last step of the cross is selfing in F1 plant to produce F2 generation. Okay, so we know that in case of monohybrid cross, two different type of gametes are produced by F1 generation plant. But now in this case, as we are considering two pair of characters simultaneously, total four type of gametes are produced by this F1 generation plant. Okay, so now capital Y, small y, and capital R, small r is the F1 parent, and this F1 parent produce four different type of gamete. This capital Y can go with capital R. Now this capital Y also combined with small r. So these are the two different type of gamete produced by using this capital Y. Now the small y can go with capital R, and small y can also form gamete with small r. So F1 hybrid or F1 dihybrid in this type of cross produce four different type of gametes. And to find out the phenotypic and genotypic ratio of F2 generation, we have to use Punnett square. Okay, because there are four different type of gametes which is produced by male plant, and similar four gamete produced by female plant. So there will be total 4 into 4, 16 different type of combinations. And to find out all these combinations, we have to use this checkerboard method, also called as Punnett square method. Okay, so now I'm going to write female gametes on top and male gametes on this side of the Punnett square. Okay, so same four gamete, capital Y, capital R, capital Y, small r, small y, capital R, small I, small r on one side of the checkerboard, and similar four gamete will be produced by other parents, okay, which will be written in the other side of the checkerboard. Now we're going to see all the different type of combinations which is produced in F2 generation. Okay, so first this capital Y, capital Y, capital R, capital R, capital Y, capital Y, capital R, small r, capital Y, capital R, and capital Y, capital R, small r. We have to write capital letter first in every combination. Now in this case, capital Y, capital R, small r. Now capital Y, capital R, and capital Y, capital R, small r. Now here capital Y, small r. Now capital Y, small r, capital Y, capital R, small r. Now capital Y, capital R, small r. Then small y, capital R. Then the small y combined with the small y and capital R, small r. And in the last three, capital Y, small r, small y, capital R, small r, and small y, small r. So 14 different type of combinations of the allele occur in F2 generation. Okay, and now on the basis of this Punnett square, we can easily find out phenotypic and genotypic ratio of Mendel's dihybrid cross. So phenotypic ratio of F2 generation become, if the parent have single capital Y, the color of the seed will going to become yellow, and if the parent have single capital R, due to dominance, the shape of the seed will going to become round. So here the seed shape will be yellow and round, capital Y and capital R, so yellow and round, capital Y, capital R, yellow and round, capital Y, capital R, yellow and round. So first four plant have seed with yellow and round type of seed. Okay. Now the next four plant, for example, have capital Y, capital R, yellow and round type of seed, yellow and round type of a seed, and yellow and round type of seed. Now this capital Y, capital R, so yellow and round type of seed, and capital Y, capital R, yellow and round type. So out of 16, 9 plant have yellow and round type of a seed. Okay, so phenotype will be 9:2. Now this plant have capital Y, but both the R's are small. So the seed shape will going to become wrinkle, but the color will be yellow. So yellow and wrinkle. Similarly, this is yellow and wrinkle because I have one capital Y, and yellow and wrinkle. So these three plants, 1, 2, and 3 will going to produce yellow and wrinkle type of a seed. So 9:2, 3. Then remaining, out of remaining 4, this plant have one capital R. So it will going to produce green and round type of a seed because both the Y's are small. So the plant will going to produce green type of the seed color, but they have single capital R, so the shape will be round. So the plant will going to produce green and round type of a seed. Similarly, this will be the plant with green and round type of a seed because single capital R, and this plant also produce green and round type of a seed. So out of the 16 plant, 3 plant have green and wrinkle type of a seed, while the remaining 1 plant have all 4 small letters. So it will be the green and wrinkle seed. So the phenotypic ratio become 9:2, 3:2, 3:1. Remember that the phenotypic ratio of dihybrid cross is 9:2, 3:2, 3:1. Now, if you want to calculate genotypic ratio of this F2 generation of the dihybrid cross, then you have to use a simple formula. We can determine genotypic ratio on the basis of this Punnett square, but it is time consuming process. So there is a simple formula which you can use to find out genotypic or phenotypic ratio of the any generation of the Mendel's crosses. Okay, so now the formula is we know that in monohybrid cross, this type of cross, phenotypic ratio is 3:1, while genotypic ratio is 1:2, 2:1. So to find out the phenotypic and genotypic ratio of any generation, we have to just use this phenotypic ratio of monohybrid cross 3:1 raised to n and 1:2, 2:1 raised to n. This n is nothing but the number of characters that we select in a particular cross. For example, in this cross we have select two characters, seed color and seed shape. So if I want to determine phenotypic ratio of the dihybrid cross, I am going to use this monohybrid cross ratio 3:1 into a raised to n, that is raised to 2, because we have selected two characters. And if we want to determine genotypic ratio, then we are going to use 1:2, 2:1, that is genotypic ratio of monohybrid cross as to 2. This 2 is for dihybrid cross. Similarly, if we want to find phenotypic and genotypic ratio of trihybrid cross, the 2 become, or 2 is replaced with 3, because in case of trihybrid cross we select 3 characters at a time. Okay, so now if we consider this 3:1 into 2 for the phenotypic ratio, now you can find a monohybrid cross phenotypic ratio on the basis of this 3:1 multiplied by 3:1, because this 2 is nothing but the 3:1 multiplied by 3:1. So 3 into 3, 9, 3 into 1, 3, 1 into 3, 3, and 1 into 1, 1. Okay, so now if we use this 1:2, 2:1, which is the genotypic ratio of monohybrid cross, and we use 2 instead of n, that means we are multiplying 1:2, 2:1 into 1:2, 2:1. Okay, now the genotypic ratio become 1 into 1, that is 1:2, 1 into 2, 2 into 1 into 1, that is 1. That means we have to multiply this 1 with all 3 numbers, this 2 with all 3 numbers, and then this 1 with again all 3 numbers. Okay, now if we multiply this bracket with 1, the result will be 1:2, 2:1. Then if we multiply the bracket with 2, then 2 into 1, that is 2, then 2 into 2, 4, and 2 into 1, 2. And finally, if we multiply the bracket with 1, it will going to become 1:2, 2:1. So this is the genotypic ratio of the dihybrid cross of the Mendel. We can simplify it as 1:2, 2:2, 2:1, 1, 2, 2, 1, as to 4, then again 1:2, 2:2, 2:1. Okay, instead of this, we can write genotypic ratio like this: 1, 2, 2, 1, in middle 4, and again 1, 2, 2, 1. Okay, so this is the genotypic ratio of the dihybrid cross. Okay, on the basis or by using this formula, you can calculate the genotypic and phenotypic ratio of trihybrid cross. So this is all about the dihybrid cross of the Mendel. It is the cross in which Mendel studied inheritance pattern of 2 characters at a time, that is seed color and seed shape. Yellow is the seed color, green is the seed color, while the round and wrinkle is the seed shape. In F1 generation, Mendel got all yellow and round. Now in F2 generation, he was expecting 75% yellow round and 25% green wrinkle, but to his surprise, he got 4 different type of combination. That is 9:3, 3:1, yellow round, yellow wrinkle, green round, green wrinkle. And this has happened because of independent assortment. That means both the character yellow or seed color can be independently assort. So yellow can be combined with round, yellow can also go with wrinkle, and green color also go with round, and green color also combined with wrinkle. So in F2 generation, Mendel got 4 different type of combination which can be easily studied with the help of Punnett square. The phenotypic ratio is 9:3:3:1 and genotypic ratio is 1:2:2:1:4:1:2:2:1. Thank you.